Recumbinant DNA technology and molecular methods. Cloning technology. It means generation of many copies of DNA, for example, recombinant DNA by replication in a host. Developed in the 70s, can generate unlimited supply of gene copies, many applications. For example, genetic mapping, DNA sequencing, mutation studies, transformation, genetic engineering. Recombinant DNA libraries, restriction enzyme site analysis, analysis of gene transcript, what is being expressed, polymerase chain reaction, DNA sequencing to be included. DNA cloning overview. The goal is to generate large amount of pure DNA that can be manipulated and studied. DNA is cloned by the following steps: isolate DNA from the organism, extract DNA, cut DNA with restriction enzyme to a desired size, splice or ligate each piece of DNA into a cloning vector to create recombinant DNA molecule. Cloning vector, artificial DNA molecule capable of replicating in a host organism, for example bacteria. Transfer recombinant DNA, cloning vector, DNA fragment into a host that will replicate and transfer copies to a probe. Step 1. Isolate the whole genomic DNA from the organism. DNA extraction easily performed using detergent to break up cell membrane and organelles, salt, sodium chloride, lyse cell and bind the DNA strand together, proteinase to digest protein bound to DNA, ethanol to precipitate and wash DNA, water to resuspend and store DNA. Storage: DNA can be stored short-term at room temperature, but it's best stored long-term at -80 centigrade or in liquid nitrogen. Ideal storage condition: large uncut DNA fragments. Suboptimal storage condition: many small DNA fragments. Average size of the DNA fragment is important for application involving large regions of DNA sequence, less important for the application involving short region of DNA sequence. Step 2. Cut DNA with restriction enzyme. What are restriction enzymes? Restriction enzyme recognize specific base pair sequences in DNA called restriction sites and cleave the DNA by hydrolyzing the phosphodiester